I'd share.
Okay.
Thank you. No, you're welcome to go ahead. I just wanted to welcome everyone over and thank you all for jumping over. We may go just a couple of minutes into the break since we're starting a little late, but Dr. Iwasawa, I will turn it over to you.
Thank you. Thank you so much for your introduction. Hello, my name is Kentaro Iwasawa. Please call me Ken. Ken is okay. I'm a research fellow in Dr. Takanori Takebe's lab in Cincinnati Children's. I'm doing some collaborative work with Dr. Stacy Herbert too. Today I'm going to talk about my project, modeling human hepatobiliary pancreatic organogenesis from the foregut midgut boundary. So just another confirmation, you can see my slides and hear me correctly?
Yes,
oh,
it looks
okay,
good.
Good.
Looks good.
Okay, so the hepatobiliary pancreatic, this is liver, the bile duct, and the pancreas. So I'm going ahead. So I'm working with pluripotent stem cells and organoids. So what are organoids? Organoids are 3D structure derived from stem cells that recapitulate the native tissue architecture and function. So in the early 2000s, Dr. Shinya Yamanaka, he found that the technology of inducing pluripotent stem cells, iPSCs, from a somatic cell from human. So any fibroblast from skin or from blood cells, using that somatic cell and reprogramming those cells, we are able to produce these pluripotent stem cells that can differentiate further into epithelium progenitors, mesenchymal progenitors that we were a little bit talking about in the previous session, and also endothelial cells, progenitors also, which we were talking in the very first session today. And using this knowledge of development, the cues, we are also able to use these progenitors to further differentiate into hepatocytes, the liver. And also the liver consists of multiple cell types, and using this co-culture system, we are also able to develop similar, this is also called human liver organoids, but with a lot of other cells as well. And using this human liver organoids, we are able to apply them and study the early development, which I'm focusing about a little bit today, and the organogenesis, but also modeling diseases such as Alagille syndrome, and also using them for drug screening to see, find a good drug, especially in the future for personalized medicine, and also to regenerative medicine. And but our human liver organoids, although they consist functionally developed hepatocytes, they completely lack the bile duct integration. Although they do consist a little bit of cholangiocytes, which are the components that make the bile duct, but they don't quite form this bile duct that can excrete the bile acid produced from these hepatocytes. And this lack of biliary integration results in poor survival, especially in long term. This problem issue that we're facing with the human liver organoids is quite similar to what we're facing in the Alagille syndrome patient livers. So the paucity of the intrahepatic bile duct in Alagille liver, Alagille syndrome patient's liver, the accumulation of the bile acid, and that causes liver injury. So in both aspects, the need to integrate the biliary system into our liver organoids is the need in the future applications, as well as studying the organogenesis. So to approach this problem, we went further back into the development, and using our knowledge that the foregut midgut boundary, which I'm going to talk about a little bit, are the origins of the hepatobiliary pancreatic progenitors, and to develop these human organoids, which consists of all the liver, the bile duct, and the pancreas with interconnections. And aim 3 would be to model a gene, recapitulate the in vivo findings in this in vitro deep organoid system. So in first, I'm going to talk a little bit about the development of the liver, bile duct, and the pancreatic progenitors. These arise from the boundary of the foregut, the anterior gut, and the midgut, the posterior gut. So as you can see here in this schematic, the liver, the bile duct, and the pancreas, these are very early progenitors that just arise from the boundary of the anterior gut and the posterior gut. And it is known that the anterior gut have an expression of this gene named SOX2, and this posterior gut is have a gene expression of CDX2. And using our knowledge of the development, we were able to induce the iPSCs, the pluripotent stem cells, and using these signaling pathways, we were able to differentiate these into this anterior gut, which have a high SOX2, and posterior gut, which have a high CDX2 expression. And this is what the cells look like in real images. So these anterior gut spheroids were produced, and they have SOX2 marked by red in this illustration, in this image here, and also with gene expressions and cell surface marker expressions. And on the posterior gut, you have CDX2, which is marked by green, and also showed in similar analysis. And when we fuse these two spheroids together, meaning just putting them adjacently, they fuse spontaneously by the next day. And the boundary formation, we were able to do this boundary formation in over 90% of the spheroids that we produced. And this boundary would be the boundary of SOX2 and CDX2, recapitulating the development. And we were now further going on for a little bit long-term culture to see if the hepatobiliary pancreatic progenitors arise from this boundary. And that's what we exactly saw. So again, this is the anterior and the posterior gut marked by SOX2 and CDX2. But as the time goes with the boundary formation, you can see the PDX1, which is a pancreas / extrahepatic bile duct progenitor marker, and also HEX, which is a hepatoblast progenitor marker that spontaneously arises from the boundary of the two. And this is this happened spontaneously without adding any signaling cues to the media. And we also found that the fusion of the anterior and the posterior gut spheroids is essential for these gene expressions to arise. So when it is not, the two spheroids are not fused, the PDX1, the pancreatic progenitor genes, and HEX are not expressed. They are not expressed. They're almost the same level as the day 8 spheroids before fusing. But when it's fused, the PDX1 and the HEX1 expression is increased. As you can see on this staining here, in the boundary, you see the PDX1 and the HEX expression coming out. And that was also confirmed using RNA, which is looking at a lot of other genes that are related to these hepatobiliary and the pancreas, and also the foregut and the midgut expressions. And you can see that the boundary are expressing a lot of these hepatobiliary pancreatic genes. And further long-term culture allows this invagination, what we call invagination of these hepatobiliary pancreatic progenitors. And so PROX1 is another gene that is a marker for all the the liver, the bile duct, and the pancreas progenitors. And we gene edited this iPSC so that the PROX1 expressed cells express red fluorescent TD tomato um, uh, in, in, in the iPSCs. So when we when you see the day 14 boundary organoids, you see the PROX1 expression again rising from the boundary, and as the time goes, you see this invagination, the proliferation of this hepatobiliary pancreatic progenitors from the boundary. This is quite recapitulating the development. And this is like day 40 to day 60 organoids. And we were able to find interconnections via biliary-like structures in these organoids. So this invaginating liver portion and the bile duct portion and the pancreas with the intestine. And this high magnification here. AFP is also a liver progenitor marker. A little bit immature, but you see this liver portion that is connected to this bile duct-like structures marked by EpCAM. These are also pancreatic regions as well in this same organoid, and you see this pancreatic region connected with DBA, which is a bile duct marker, and also in here the PROX1 is also connected with DBA marked cells. So in these from these data, we were able to develop a foregut midgut boundary model that developed human hepatobiliary pancreatic organoids with interconnections. And our aim 3 was to model the HES1 mediated organ segregation erroring in HPP organoids. I'm talking about this HES1. Why am I talking about this HES1? So we know that the Alagille syndrome patients have mutations a lot in JAK1 and also a few in Notch2. This JAK1 is, as you all know, the singly sending the ligands, and Notch2 are the receptors. And further downstream of this is a target protein that is produced from this signaling is a protein called HES1. And Dr. Stacy Herbert mentioned this paper briefly in her presentations, but it was shown that this when the HES1 deficient mouse is produced, this HES1 deficient mice, we can see a conversion of the biliary system to pancreatic tissue was observed in this HES1 deficient mice. So in other words, the biliary system is diminished and the pancreatic tissue is increased. So we went back to our organoid model and to see if we are able to see a similar phenomenon in our organoids as well. So gene editing the iPSCs is a little bit time consuming, but is possible, doable. So what we did was not diminishing the HES1 from the iPSCs itself. And this HES1 plus is a wild type, so they have normal HES1 expression. And when you look at the normal HES1 expression organoids, you see the bile duct and the pancreas marked with red and green here. And you see the green interconnected with pancreatic tissues. Whereas when we knocked out the HES1 completely, the similar organoids is lacking the bile duct structures, and the PDX1 expression is increased, as you can see also in this high magnification size here. So we were able to develop anterior posterior boundary that expresses the hepatobiliary progenitors, and also recapitulating the in vivo findings in our in vitro organoids. So in the future, we have the current ongoing project that we are doing in collaboration with Dr. Stacy Herbert, is that we have generated Alagille syndrome patient-derived iPSCs. So using the hepatobiliary pancreatic organoid, the model that I just showed you, and this ALGS iPSC, we're planning to further investigating modulators to improve the bile duct formation. So with that, I would like to thank my lab for their support. And I'm also going to show you let me see. I have a video of what I just talked to you, a summary video. So as I talked to you briefly, day 7, SOX2, spheroids. These are anterior SOX2 and the posterior CDX2. When we make these spheroids and combine it together, the boundary hepatobiliary pancreatic precursors arise spontaneously without any developmental cues that we add into the media. And then by time these hepatobiliary pancreatic progenitors invaginate, mimicking the developmental, recapitulating the development, and the PROX1, SOX9, and PTX1 gene expressions are seen in the organoid. And it forms the liver, bile duct, and the pancreas. And we call them hepatobiliary pancreatic organoids. And when we add the bile acid with the fluorescent, although I didn't show you this data in my presentation, but we were able to see the interconnections and the draining of this bile acid, fluorescent labeled bile acids into the intestinal structures. And in the last one, I showed you that the HES1 knockout diminishes the biliary and replaced with the pancreatic tissues. So this is the summary slide, summary video of my talk that I just presented. Thank you all for your patience, and I'm happy to discuss.
Thank you so much, Ken. We need to go ahead and close this, but if you are around, any questions can either come through me and I'll forward them to you and connect, or someone can connect with you through the lobby of that platform.
Okay.
Would that work?
Yep.
Okay, great. Thank you so much. This was really exciting. I'm so just thrilled at all of this work, and we're so happy that you could be a part of this. We are going to take a break for a little less than an hour and meet back here at 12:45 for a transplant discussion with Dr. Kamath and Dr. Lin. And again, if there are any questions, you can either send those to me and I will connect you with Ken, or just connect on the platform. Thank you again so much.
Thank you so much.
We'll
Thanks, Ken. Thanks, Ken.
Bye.
Thank you.
Thanks.